So let's talk about the synthesis of these new strands of DNA. Now we know that the strands of DNA are anti-parallel. That means they run in opposite directions. The orientation of the sugar phosphate backbone can only be created in a particular direction. So because of that, DNA polymerase can only add those nucleotides to that 3' carbon on the deoxyribose sugar. So because of that, if you're going to make a new DNA strand, we say it can only be created in the 5' to 3' direction. So again, that's just because to add another nucleotide, you can only attach that phosphate to the 3' carbon of the existing nucleotide. Now that creates two different strands, the first of which is called the leading strand. And the significance of the leading strand is that it is continuously made, so it's just one big long strand of DNA that gets made as the DNA polymerase moves toward the replication fork. So if we look here we have that replication bubble. Here's our origin of replication, and then what happens is you have that RNA primer laid down, so there it is right there. Then DNA polymerase slides along that molecule, again going from the 5' location to the 3' location. So DNA polymerase slides toward the replication fork we see here, adding these A, T, C, and G nucleotides. So there we see the new strand of DNA being synthesized. Again, the bases, or the nucleotides that are being added, are added to the 3' end of the molecule. Now, this other strand is then what is called the lagging strand. So the leading strand is synthesized continuously, but what's interesting about the lagging strand is that it is synthesized discontinuously. So we see that it's actually just a series of fragments that get added. So again, it is required to go in the 5', we'll do this here, 5' to 3' direction, which is opposite that of the other strands since they are anti-parallel, and that leads to some interesting results. This lagging strand is fascinating because, again, DNA polymerase is moving away from this replication fork, but still in the 5' to 3' direction. So what ends up being created are what are called these Okazaki fragments, named after the individual who led the team that discovered those individual segments on the lagging strand. Again, DNA polymerase adds nucleotides to form a new strand, but it does so discontinuously, making these Okazaki fragments. And then all these individual segments are stitched together using another enzyme called DNA ligase. Now this makes a little bit more sense if we look at what's going on here. So we know that primase lays down this short little primer, again 5' to 3' direction. Now this is RNA, but the sweet deal there is that it creates this 3' carbon that's sticking out. So now DNA polymerase, this is actually called DNA polymerase 3. DNA polymerase 3 can attach that 3' carbon and start adding DNA nucleotides and making a new strand, this Okazaki fragment. Now, other RNA primase or other primase enzymes will lay down other fragments or other primers, and then DNA polymerase 3 can go and attach to, again, that 3' carbon, lay down another Okazaki fragment. And then what will eventually happen is these fragments need to be replaced, right? So something needs to get rid of these RNA fragments and replace them with DNA. And that molecule is actually another DNA polymerase. This is a molecule called DNA polymerase 1. Okay? So DNA polymerase 1 cuts out the RNA nucleotides from these primers and then replaces them with DNA nucleotides. So you now have a molecule that's all DNA. The issue becomes, though, there are fragments then that have to be stitched together. So that is where ligase comes in. So ligase is the enzyme that actually stitches together the Okazaki fragments to create one continuous strand of DNA. So DNA synthesis using the leading strand is fairly simple. You lay down the primer, you then attach DNA polymerase, and off it goes, making that strand. But again, the lagging strand does add the molecules discontinuously. You have the RNA primase come in, creation of the Okazaki fragments. Then you have DNA polymerase II come in and replace the RNA fragments with DNA, and then ligase finally stitch it together. So it's a whole host of proteins working together to create this new strand of DNA. This again just sort of goes into some further detail. You can peruse that at your convenience. Please do be very familiar with all these terms. All these enzymes play an important role in the process of DNA replication, and you'll need to be familiar with their role and how to identify them and what they do on diagrams. So please make sure you're comfortable with that. This here is a nice image because it sort of ties everything together. Here we have that leading strand that's synthesized continuously. There was a primer that was laid down early on here by this primase enzyme. Then DNA polymerase comes into the 3' carbon, and it's off and running, adding A's, T's, C's, and G's, these nucleotides that were floating around in the nucleoplasm, or the fluid inside the nucleus. On the lagging strand, again, the primer lays down the original base, our primase lays down the original sequence, then DNA polymerase III comes in, adds the sequence along these Okazaki fragments. Then DNA polymerase I comes in and replaces the RNA sequences, so they go out, and they are replaced with DNA sequences. And then finally, DNA ligase stitches together or covalently bonds the sugar phosphate backbone so that you now have one continuous strand. This is, it just takes a little getting used to. Again, the key there is to walk yourself through the process. Think about how all these parts need to work together to ultimately create two new strands. Keep the big picture and the end in mind, and just look at how the individual pieces work together to make the process work, and it's much more manageable. All right, let's see. Now, these proteins that are all involved are more than likely stationary, actually. They're going to be attached to fibers within the nucleus. So there's this nuclear matrix of protein fibers, the microtubules and microfilaments, and those proteins in this replication complex are probably attached to it. So they actually reel in the parental DNA as they synthesize the new strand. And again, this is all emerging research, so maybe you could have a role in getting better detail so you can add a couple pages to the textbook for future AP students. Now, what's really cool is when mistakes happen. Well, I guess it's not cool, but it's fascinating how mistakes are detected and removed. Now, occasionally, some bases can join covalent bonds with one another. This is what's called dimerizing. So normally these bases should hydrogen bond with their partners, but occasionally errors can occur when they actually link up and form bonds with one another. Now, this is an important sequence to know. First, what will happen is an enzyme called nuclease will go and excise or cut out that segment of the DNA that contains this error. So again, this enzyme goes and cuts at specific locations and removes the damaged DNA. Then DNA polymerase will again, this is DNA polymerase III, DNA polymerase III will come in and, again, beginning with the 3' carbon, go in, add bases in so that you now have the correct sequence, and then ligase will do its job and then stitch together or covalently bond the sugar phosphate backbones of the new segment and the existing segment, so now you have repaired DNA. Very, very cool. Now, there are different types of repair that occur. Again, DNA polymerase acts as an actual proofreader. It can go and fix these bases through a process called mismatch repair. So it can actually take bases that are incorrect, excise them or cut them out, and then replace them with the appropriate base. Let's see. That said, throughout our lives, there are going to be mistakes that occur in our DNA. Exposure to chemicals, X-rays, even ultraviolet light in our skin, that's what leads to this dimerization of the thymines. That can all damage DNA. So we need the ability to cut out mistakes or damages in the DNA and then replace them with the appropriate sequence. Our ability to locate and remove errors in the DNA are significant in our continued health. So very, very cool process. Let's see. That's that for now.